IB Bio Cell Division Meiosis Part III is a short movie that wraps up some lingering concepts. The first concept is that different species have different numbers of chromosomes. The second concept examines the meiotic error known as non-disjunction. Non-disjunction results in chromosome abnormalities, and we will analyze karyograms to assess the presence of chromosome abnormalities. The essential ideas are chromosomes carry genes in a linear sequence that is shared by members of a species, and secondly, alleles segregate during meiosis, allowing new combinations to be formed by the fusion of gametes. Karyograms display the chromosomes of an organism in homologous pairs of decreasing length. Humans have 46 chromosomes total, 23 pairs, as you can see here. Now, genes are sequences of nucleotides that typically code for protein. Genes exist in specific locations, known as loci, along chromosomes. The unique staining pattern of each chromosome pair hints at the loci of specific genes. The number of chromosomes is a characteristic feature of members of a species. As you can see in this slide, humans have 46 chromosomes, while the chimpanzee, Pan troglodytes, has 48, while the domestic dog, Canis familiaris, has 78. Rice, Oryza sativa, has 24 chromosomes, and the worm, Parascaris aquarum, has just 2 chromosomes. Remember that prokaryotic cells have a single loop, with numerous tiny rings of DNA in the cytoplasm, known as plasmids. Here is a slide that compares human chromosomes with chimpanzee chromosomes. The slide displays just the haploid number of chromosomes of each species, 23 chromosomes for humans and 24 for chimps. Once again, the number of chromosomes is a characteristic feature of members of a species. In this slide, we can see humans with 46, tobacco with 48, and the fruit fly with 8. Now let's shift gears to examine the meiotic error known as non-disjunction. Here are the relevant IB syllabus statements. Explain non-disjunction that leads to changes in chromosome number, illustrated by Down syndrome, trisomy 21, and one other abnormality. Analyze a karyogram to diagnose Down syndrome in humans. The processes of meiosis do not always function as they should. Errors occur, and one of those errors is known as non-disjunction. Non-disjunction is when homologs do not separate during anaphase 1 of meiosis, or chromatids do not separate during anaphase 2 of meiosis. In both situations, one gamete ends up with an extra chromosome, while another gamete ends up with one less chromosome. Trisomy 21, also known as Down syndrome, is an example of non-disjunction. Trisomy 21 is a condition where an individual's cells have 3 chromosomes instead of 2 of the 21st chromosome type. Let's look at the next slide. Here is a karyogram that shows the chromosomes of an individual with 3 chromosomes here rather than 2. The events that produce trisomy 21 will be coming up. But in order for an individual to have 3 chromosomes 21, they receive 2 21 chromosomes in one gamete and 1 21 chromosome in the other gamete. Non-disjunction during meiosis results in the formation of a gamete with 1 extra 21 chromosome. Then fertilization results in a zygote with 3 of the 21st chromosome type. In this image, we can see the error occurring during anaphase 1, where the proper separation of a homologous pair does not occur. This cell, at the end of meiosis 1, has an extra chromosome. And after meiosis 2, we have gametes with an extra chromosome and some gametes with one less chromosome. If the gamete, either of these two, with an extra 21st chromosome gets fertilized, trisomy results. Trisomy 21, also known as Down syndrome, occurs by non-disjunction followed by fertilization. Here is a diagram that displays non-disjunction during meiosis 1 or meiosis 2. On the left, you can see non-disjunction occurring during anaphase 1, where the homologous pairs do not segregate properly. On the right, you can see non-disjunction occurring during anaphase 2, where sister chromatids are not separated. The gamete formed in either situation will have an extra chromosome, and following fertilization, trisomy will result. Here is a karyogram of a human with trisomy 21, someone with 47 chromosomes rather than 46. You can see the 3 chromosomes of the 21st type here. You can see that the person from whom these chromosomes were extracted was a female with 2 X chromosomes. Here is a karyogram of an individual with 1 less chromosome, 45 instead of 46. There is no Y chromosome and only 1 X. This situation also results from non-disjunction, but it results from the non-disjunction of the sex chromosomes. This condition is known as Turner syndrome. Remember that non-disjunction results in some gametes with 1 extra chromosome and some gametes with 1 less. Turner syndrome occurred when the cell with 1 less chromosome was fertilized. Here is a karyogram of an individual with 1 additional chromosome, like trisomy 21, but the extra chromosome here is an extra Y chromosome. There are 3 total sex chromosomes. This condition is known as Klinefelter syndrome. Here are the IB syllabus statements that will bring this movie to its end. Describe the methods used to obtain cells for karyotype analysis, including chorionic villus sampling and amniocentesis. State the role of chorionic villus sampling and amniocentesis. Describe the risks associated with chorionic villus sampling and amniocentesis. I will not spend a lot of time with any one of these 3 statements. Thus, you may want to take the time to study the remaining slides of this movie. Chromosome abnormalities such as Down syndrome or Turner syndrome can be detected before birth by a technique known as amniocentesis. Fluid is withdrawn from the amniotic sac of the fetus. This is done prenatally. The amniotic fluid contains fetal cells. The cells are cultured into dividing by mitosis, where the chromosomes become visible due to condensation during prophase of mitosis. A technician makes a squash of the cell just before mitosis occurs and then examines the chromosomes by building a karyogram, as I've shown you. The karyogram is analyzed to provide a karyotype that is assessed for the presence of chromosome abnormalities. Chorionic villus sampling is another technique that provides information on possible chromosome abnormalities. A catheter is inserted into the uterus through the vagina, and a tissue sample is taken from a location within the placenta known as the chorion. Fetal cells are withdrawn from the chorion and cultured into mitosis. A karyogram is constructed and analyzed to provide a karyotype assessing for possible chromosome abnormalities. Chorionic villus sampling has certain advantages over amniocentesis. It can be performed earlier and the results come back more quickly. The information allows couples to make an informed choice about their pregnancy. The health risks of undergoing chorionic villus sampling or amniocentesis are the following. For amniocentesis, there's a risk of miscarriage, 1 in 300 to 1 in 500 events. There is possibility of needle injury, possibility of infection, and the infection being transmitted to the fetus. For chorionic villus sampling, there is a risk of miscarriage, carries a 1 in 100 risk. As well, there is a risk of infection. Studies have been done that suggest that the mother's age influences the frequency of Down syndrome. The risk of Down syndrome increases as women get older. Among 25-year-old women, Down syndrome occurs 1 in 1,250 births. Among women who are 40 or older, Down syndrome occurs in 1 in 100 births or more frequently. And that brings us to the end of IB Bio Cell Division, Meiosis Part III. On to genetics.